Hi everyone, this will be a faceless video on the central dogma, and we will be starting with DNA replication. As a review, here's your central dogma, which shows us the relationships of the three biomolecules, particularly your DNA, your RNA, and your protein. So now, replication refers to the duplication of your DNA. Again, your DNA contains the blueprint of life. It contains the code. This code stores the important information necessary for life. And of course, if any cell wants to reproduce or to divide, it has to create an exact copy of its genetic material. So for this discussion, we will be focusing primarily on the prokaryotic DNA replication. So with the discovery of DNA being the biomolecule that stores genetic information, of course, the next question would be how is the genetic information passed on from a parent to the offspring, right? And that's when they discovered that um, the process of reproducing a copy of a DNA molecule is semi-conservative. So, how exactly does this semi-conservative process take place? So, for prokaryotes, let's say bacteria, their DNA is typically circular, as shown here. And replication starts at sites or specific sequences in the circular DNA of your bacteria called ORI, ORI, which is short for origin of replication. So the enzymes necessary to make this process possible recognizes specific base pair sequences in the ORI, and they would know that this is the spot where they should start the process. So once the enzymes assemble here around the ORI, the replication initiates. So it starts, and it produces this so-called replication bubble. Okay, and as you can see, we have here what we call the replication fork, and this is where the reproduction of the new DNA strand takes place. Okay, and when the process is complete, we now have two daughter DNAs. One of the strands in each daughter is from the parent DNA, and the second strand for each daughter would be the newly synthesized strand that was produced during replication. So the evidence that proved that this is how DNA replication takes place in bacterial cells is through an experiment called autoradiography, where essentially a scientist can use a heavy isotope, which is also radioactive, in order to visualize any biomolecule that they wish to see. And they tried to use these heavy radioisotopes to actively dividing bacterial cells. And that's where they discovered or visualize the replication bubbles. Okay, so here you see the two replication forks, and we see there the replication bubble, and then eventually this bubble gets larger until we see the two daughter DNA getting separated from each other. So we will be discussing details on how this process takes place. So DNA replication, always remember, is a semi-conservative process. and you already know what semi-conservative means. So DNA replication begins at a site that we call ORI. So there's an origin. It doesn't occur or it doesn't start randomly along a stretch of DNA. Replication proceeds bidirectionally. There are two replication forks, and the direction by which the replication takes place at these two replication forks are opposite each other. And because we have two replication forks, we should be able to observe a replication bubble. Now, keep in mind, as you have learned, the synthesis of new DNA strands only occur in one direction. So recall that your DNA strands have directionality. So when you synthesize a new DNA strand, it's always from the 5' to the 3' direction, which means you always start with a nucleotide with a free phosphate group. And then you always end with a nucleotide that has a free hydroxyl or alcohol group in its 3' carbon. And this is something new. DNA replication is considered semi-discontinuous, and we will later on see why this is so. And of course, the enzyme responsible for DNA replication, they are called DNA polymerases. But before that, let us remember that if there are DNA polymerases which synthesize DNA, there are also enzymes that degrade DNA, and we call them DNA nucleases. And we have two types of nucleases, endonuclease and exonuclease. So, like what you have learned about your enzymes that degrade proteins, when we say endonuclease, these are enzymes that cleave or cut DNA strands in within a DNA strand, versus an exonuclease. This is an enzyme that can cleave only the terminal nucleotide. So it can be a 5' nucleotide. So it's the nucleotide that you will see in the 5' end of a DNA strand, or it can be a 3' exonuclease. So what it can cleave is the nucleotide in the 3' end. So knowing that these enzymes exist will help us in the understanding of DNA replication. Okay, so here is a very good diagram of what exactly a replication bubble looks like and what is happening here. So we will see here the important molecules involved in replication, such as these topoisomerase, helicase, etc. The only thing you have to note at this point is the features. So you have the replication bubble and you have the two replication forks. One of these replication forks is going from right to left, and the second replication fork is going from left to right. And what I mean by this is the synthesis of the new DNA strand. Okay, but the way the replication fork itself is growing, so here you see this arrow from left to right. So at this point, the size of the bubble increases as this replication fork migrates from left to right. But the synthesis of the new DNA strand is from right to left. And on the other side, here on the left replication fork, so at this point, the replication bubble size will increase as the replication fork migrates from right to left. But the synthesis of the new DNA strand takes place from left to right. Okay? And of course, let me just remind you that when you synthesize a new DNA strand, it is important that you have a template strand. That's why you need the parent DNA. Both strands of your parent DNA will serve as templates. Okay, so let's focus at one replication fork. So we have the parent DNA here represented by the gray lines, and we have the pink line and the shorter blue lines representing the newly synthesized daughter strands. And this is where we see why DNA replication is semi-discontinuous. Okay, so when we say semi-discontinuous, as you can observe here, one of the new daughter strands, since its template is the 5' to 3', and you know that the synthesis of new DNA strands occurs from 5' to 3', then there is no problem here. There's no issue in the synthesis of the new strand. So it will occur continuously. Okay, versus if we are using the 3' to 5' strand of the parent DNA as the template, we cannot do the synthesis of the new or the daughter strand continuously because of the replication fork. Okay, so this is the hindrance. So what we do, or what the cell does instead, is what we call the lagging strand. So it creates short DNA strands using this 3' to 5' strand as the template. Okay, so again, with the 5' to 3' as the template, it creates the leading strand. But with the 3' to 5' template, it creates a lagging strand. So this is the other replication fork. And as you can notice, on this side, okay, what is considered as the leading strand this time is the one that is using the 3' to 5' parent strand as the template. And then the lagging strand is now produced using the 5' to 3' parent strand as template. So probably would it's probably difficult to imagine, but you can always go back to this diagram. Okay, so recall that this is our ori or origin. So this is where the synthesis of the new DNA will take place. So if you are looking at this replication fork on the left side, then this will be your leading strand. But if you are looking at the replication fork on the right strand, this will be your leading strand. Okay? So it's a matter of perspective. That is why overall we say that DNA replication is semi-discontinuous. Okay, it's not exactly completely discontinuous because we have here this leading strand. So it just continues by itself. Okay. However, if we look at this side, this will be our leading strand. Okay. So now, as I mentioned, DNA replication is catalyzed by your DNA polymerases. And if you notice, it's in the plural form. That is because for any organism, it possesses more than one DNA polymerase. But among all of these DNA polymerases, only one of them will be the main polymerase for DNA replication. The other DNA polymerases are used for proofreading. So they are the ones that check if the correct nucleotide is paired, and then they can perform some corrections. So for the E. coli, there are three well-established DNA polymerases, but the E. coli possesses more than three DNA polymerases. But these three are already well known, and they are called Pol A, Pol B, and Pol C, Pol short for polymerase. And you can see the relative sizes of these polymerases. And of course, the largest would be polymerase 3. And this polymerase 3 is the main polymerase for E. coli DNA replication. So if we look at this table, there is what we call 3' to 5' exonuclease activity. If you recall, when we say exonuclease, this is the catalysis of removal or cleavage of the nucleotide in the 3' end of a DNA strand. On the other hand, 5' to 3' exonuclease activity means the enzyme can remove the nucleotide in the 5' end. Okay? All of the DNA polymerase listed here can remove the terminal nucleotide at the 3' end, but only DNA polymerase 1 can remove the nucleotide at the 5' end. So polymerase 1, its most important function is correction. Okay? And what else? We have the polymerization rate, which means how many nucleotides can this enzyme incorporate in the newly synthesized strand per unit time. And of course, since polymerase 3 is the main, it should be fast, right? So 250 to 1,000 nucleotides per second. Meanwhile, polymerases 1 and 2, they are quite slow because, again, their main function is for correction. And lastly, we have this property that we call processivity. So processivity means the amount or number of nucleotides that the polymerase can add to the newly synthesized strand before it dissociates from the replication fork. Okay, so polymerase 3, again being the main polymerase for replication, has the highest processivity, and the other two, they have low processivity. And again, this is a reflection of their functions. Okay, so the mechanism for the synthesis of a new DNA strand. So this is in the active site of your DNA polymerase, generally speaking, because again, we have lots of DNA polymerases. We have, of course, magnesium ions. Magnesium ions here help stabilize the highly negative charge of your triphosphates or your nucleoside triphosphates. And these magnesium ions are also stabilized by negatively charged amino acid residues in the active site. As you can see here, you see aspartate. Okay, so magnesium ions, aside from stabilizing the highly negative charge of your nucleoside triphosphate, it also drives the removal of proton from the 3' hydroxyl group of your DNA strand. Okay, And with this, it becomes more nucleophilic and can now attack more efficiently the gamma phosphate. Okay, so alpha, this is the farthest from the sugar ring. So this is the alpha phosphate, beta, gamma. So it attacks the gamma phosphate, and then you release the diphosphate, which is also called pyrophosphate, and you now have a new nucleotide attached to the growing DNA strand. Okay, so how is the base pairing ensured? So you know that A will always pair with T and vice versa, and G will always pair with C. So, of course, we are already familiar with the fact that this pairing has the optimum number of hydrogen bonds. But when in the context of DNA replication, aside from the optimized hydrogen bonding between AT and GC, the shape of the active site in the DNA polymerase also assists in the correct base pairing. As you can see here, the shape of the active site is outlined, and you can see how each base pair fits into the active site. And if you try to place like in an incorrect base pair such as GT, it doesn't fit in the active site. Okay. So when we talk about DNA replication, again, its primary objective is to create an exact copy of the organism's gene. So nature makes sure that replication is replication has high fidelity. So when we say fidelity, this is the room for mistake. So there should be low frequency of mistakes, of course, because if the frequency is very high, then that would lead to more mutations and, of course, more diseases for the organism, less chances of survival. So, on average, this is again in the context of prokaryotic DNA replication. So, on average, there is one mistake in 1 raised to 9 to 1 raised to 10 nucleotides added in a new DNA strand. But this number, again, this is the performance of your DNA polymerase 3. But in reality, like if you encounter a mistake, so this mistake could probably occur like one incorrect nucleotide for every 10 raised to 4 to 10 raised to 5 correct nucleotides. So that is the average performance of prokaryotic DNA replication. Now, interestingly, these figures, I think, because of course, if the process still makes mistakes, then as years go by, the organism should no longer exist, right? Because the its DNA sequence has been like changed. But interestingly, we still have E. coli in existence now, right? Like from years back when it was only the prokaryotes existing before the eukaryotes evolved, these prokaryotes are already existing. So it's not the eradication of the organism, or shall we say extinction of the organism, but it's more of you get a new organism probably. Anyway, I'm just yapping here. Okay, so let's proceed. Okay, so I mentioned that DNA polymerase 3 is the main polymerase for replication. The rest of your polymerases are for proofreading. DNA polymerase 3, as we saw, is a very large enzyme. It has multiple subunits, and these subunits are designated with Greek letters: alpha, epsilon, theta, tau, gamma, delta, delta prime, chi, psi, and beta. And you can also see how many of each subunit is present in the whole complex of your DNA polymerase 3. And you can see the functions of each subunit. The alpha subunit is the main, meaning that it's the one performing the polymerization activity. It's the one that reads the template, the nucleotide from the template strand. It's the one that um, puts in the partner nucleotide. So if it reads T, it should put A, and then it catalyzes the condensation reaction. The epsilon subunit is the one that has the 3' exonuclease activity. The theta is important for the stabilization of the epsilon subunit. And then the other subunits are essential for the physical process, because you know that the replication fork should be moving, right? And it's at the replication fork where you have the synthesis. The addition of the next nucleotide takes place at the replication forks. Okay, so the DNA polymerase should move along the replication fork, and that is made possible through these other subunits. Okay, so as mentioned, the addition of each nucleotide in your growing daughter strand takes place in the replication fork. And here in the replication fork, you will find not just your DNA polymerase, but also other enzymes that help in making DNA replication possible. And this whole thing, the replication bubble itself, plus these various enzymes doing their specific functions, it's called the replisome. Okay, replisome, or the DNA replicase system. So we will go through each enzyme one by one. Again, these enzymes are different from your DNA polymerase. Okay, so the first enzyme is helicase. So the helicase is responsible for opening the double helix of your DNA, because structurally speaking, it's impossible for you to create a new DNA strand if the two strands of your parent DNA are interacting with each other. So you should open the double helical structure of any DNA molecule for replication to take place. And that is made possible through your helicases. So your helicases, they need energy. And of course, when we talk about energy inside the cell, this needs ATP, right? So it uses ATP to make this physical unwinding of your DNA molecule possible. And of course, you will find your helicase at the replication fork, because the replication fork needs to grow in size, right? Now, if you are unwinding one section of your DNA, as you can see here, it means that the rest of your DNA strand will now become more winded, if you know what I mean. If you can imagine what I'm trying to say. Okay, so imagine you have two strands that are intertwined with each other, like a rope, right? A rope is made up of several fibers intertwined, right? If you try to unwind one segment of your rope, you will notice that the intertwining in the other segments of the rope gets tighter. That's what I'm trying to say. Okay. In the context of DNA, this is what we call topological stress. Okay. So it becomes supercoiled. So you have the coiling or the double helical DNA, but because you are opening this double helical structure, the DNA itself becomes super coiled. So that introduces what we call topological stress. And we have what we call topoisomerases to relieve that topological stress. So you will see your topoisomerase a bit ahead of a replication fork, as you can see here. Okay, that's the function of your topoisomerase. Next, of course, if you are separating the two strands of a DNA molecule, you now have exposed nitrogenous bases. So because you don't have the base pairing anymore, the nitrogenous bases are exposed. And for them to be stable, because they lost their partners, right? The hydrogen bonding of the base pairs that keeps them stable is broken. So in the meantime, while waiting for the new strand, the exposed single-stranded segments in the replication fork or replication bubble are made stable via DNA binding proteins, or here you see the acronym SSB, which stands for single stranded DNA binding proteins. So these proteins simply bind to single-stranded segments to keep them stable, but they also dissociate if the polymerase approaches that region of the single-stranded DNA. Next, we have primases. What are primases? So I don't know if you already asked this question. If you already have, or if the cellular mechanism already found the origin in the DNA molecule, how is the DNA polymerase going to start? Right? Like, you saw the mechanism, the reaction mechanism. It needs that 3' OH group to attack the gamma phosphate of the incoming nucleoside triphosphate. So to answer that question, okay, like, where is that free OH group? To answer that question, we look at primases. Primases are enzymes that create short or oligonucleotides, RNA oligonucleotides, and this serves as what we call primer. So the primases, uh, they also have polymerase capability, albeit RNA polymerase capability. So they read the template strand and they synthesize a complementary RNA strand. So this is just a short RNA strand, probably like around 50 nucleotides, something like that, or probably shorter, depending on the organism. And then once the primase synthesizes the primer, that's when the DNA polymerase can start its work. Okay? Because in the primer, you have the 3', the terminal 3' there, which should have a nucleotide with a free 3' OH group. Okay? So your DNA polymerase cannot work, cannot start its function without a primer. Okay? Now you might ask, how are these RNA segments removed? We will discuss that later. Okay. And then this is now the answer. Okay. So for the removal of your RNA primers, this is performed by DNA polymerase 1. Okay. So your DNA polymerase 1 recognizes those RNA nucleotides, and it knows that this shouldn't be there in the newly synthesized DNA strand. And recall that it has exonuclease activity, but it can also synthesize DNA, right? So what it does is it it removes the nucleotides from the RNA strand one by one, replacing it with the correct DNA nucleotide. Again, reading the template. And then, yeah, until all of the RNA nucleotides are replaced. Okay? Now, the thing is, your polymerases, they cannot synthesize, they cannot catalyze the attachment of a 3' OH of one strand to the 5' phosphate of another strand, if you know what I mean. So your polymerases can catalyze. So if this is one DNA strand, you have the 3' end, right? It can catalyze the condensation of this 3' end to a deoxynucleoside triphosphate, which is just a nucleotide, a single nucleotide. But it cannot synthesize the conjugation of two DNA strands. So this is in the context of your lagging strand. If you recall, in your lagging strand, the DNA is synthesized segment by segment. Okay? So the problem here is if you have this 5', the beginning of one DNA segment, and then you have this 3', which is the last nucleotide of the previous DNA segment, DNA polymerase cannot synthesize the conjugation of these two strands. So you need a different enzyme for that. And that is what we call ligase. So your ligases catalyze the attachment of these strands. So this thing has this has a name. So we call that nick. So a nick is when a 3' end is not attached to a 5' end along a DNA strand. So nicks are resolved by DNA ligases. Okay? So you also need DNA ligases during DNA replication. And now we didn't miss anything, right? So we have the helicase, topoisomerase, primases, ligase. Okay. We now go to the steps or stages of replication. Okay. We now have a general idea of how replication takes place, but at each stage, there are important things that we have to know. These are the stuff that makes replication possible. So we start with initiation. So there are only three stages: initiation, elongation, and termination. So with initiation, the important thing here, well, there are three important things. Number one is the recognition of your ori. So how does a cell know that that DNA segment is the ori? That's where the replication bubble should form. So they performed this experiment called denaturation mapping, and you can read it. There's a short description of this experiment in Leninger, denaturation mapping. And they found out that these origins or ori in prokaryotic DNA are rich in adenine and thymine nitrogenous bases, or AT rich. And if you recall, the AT base pair has only two hydrogen bonds compared to your GC. And it makes sense that you should have an origin that is AT rich because it requires less energy to open. So again, you need to destroy the hydrogen bonding because you're going to open the your DNA. You have to break uh, the interaction of the nitrogenous bases for you to form the replication bubble. So they called these ori, or these the base sequence found at the ori. They call it. Let me just type that. DUE, short for DNA unwinding elements. If I'm not mistaken, yeah, it's DUE or U, DNA unwinding elements. So this is the specific sequence where you have AT rich segment. Okay, so the whole thing, as you can see here, is labeled ORIC. So that's your point of origin, or region of origin for DNA replication. But you have DUE specifically because this is where the unwinding of your double stranded DNA will take place. Okay, it's the DUE which is rich in AT. It's different from your ORI. Your ORI has ORI, or replication origins, also have defined um, base sequences or DNA sequences. DUE is AT rich because this is where the unwinding will start. Okay, so the first step that will take place here is the uh, loading of your DNA A. So we have here DNA A, which are ATPases. So when you see the term ATPase, these are enzymes that catalyze reactions requiring energy. So they need ATP. They will get energy from ATP to be able to catalyze the reaction that they are supposed to catalyze. So DNA is an ATPase. And these DNA molecules or enzymes are the one responsible for the initial unwinding of a double-stranded DNA in the origin. Okay, it's not helicase. Helicase is responsible for the unwinding at the replication fork. But when you initiate replication, you need these DNA A to unwind the DNA around the DUE, or the DUE, DNA unwinding element. So in particular, for E. coli, it needs 8 of these DNA A enzymes. So the DNA A enzymes will um, wrap themselves with the DNA and catalyze the unwinding. Okay? So as you can see here, the term is denaturation because you are trying to break the interaction of the two strands of your DNA. Now, once this is achieved, the next step would be the loading of your helicase. So we have here DNA B, which is a helicase, and DNA C. DNA C acts like a delivery molecule. So its purpose is just to deliver the helicase. Okay, so it should deliver 2 helicases for each replication fork. Okay, so there should be 2 DNA C. One DNA C is attached to one DNA B. So since you have 2 replication forks, 2 of these complexes will enter the picture. Okay, and then the DNA C, again using the energy from ATP, will load the DNA B or the helicases at each replication fork. Okay, so all of these steps are aided by other proteins as listed here, which again, we will not be dealing with too much. Okay, so we have the initial unwinding of the DNA, and then we have the loading of the helicases. I guess that's it for the initiation. Okay. Next would be the elongation. So for the elongation, this is where your DNA polymerase enters the picture. Okay. So once you have the replication bubble formed, actually, there is sort of an overlap with initiation and elongation. Okay, in initiation, this is where the rest of the replisome or the replicase assembly. Am I right? Sorry, replicase system or replisome assembles. So you have the DNA polymerase, the topoisomerase. Once you have the helicase there in the replication fork, then the others like topoisomerases, your single-stranded DNA binding proteins, primases assemble. Okay? And then we now go to elongation. And in this stage of replication, this is where your DNA polymerase stars. Okay. For this one, we have, yeah. So we just basically have the formation of the leading strand and the lagging strand. Okay, please keep in mind the figures that we have here, we are looking at the left replication fork. The story would be a bit different if we are looking at the right replication fork. Okay, so the leading strand here is here, and then the lagging strand is down here. You have the primase there, which synthesizes the RNA primers necessary for each segment of your lagging strand. Okay. Um, what's interesting here to note is that the leading strand is always synthesized in the same direction as the migration of your replication fork, while the lagging strand is synthesized in the opposite direction with that of the replication fork. That's something noteworthy. Okay. So, what is happening in the elongation stage? The thing is, from the name itself, lagging strand, does it mean that the rate of synthesis of your lagging strand is lower than that of the leading strand? Is it really like lagging in that sense? So keep in mind that the synthesis of the two new strands from the two template strands must happen at the same time or almost at the same time. And this is where the architecture of your DNA polymerase comes into play. So, again, it's the beauty of the shape, the structure of your proteins, enzymes in this case, that is fascinating in biochemistry. Okay. So, recall that your DNA polymerase 3 has a lot of subunits. You have the you have the core, which consists of the alpha, epsilon, and theta. So the alpha is the one that polymerizes DNA. The epsilon is responsible for proofreading. And then you have the theta, which stabilizes the epsilon subunit. And then you have the tau. This tau is its function is like a setter. Okay, so it setters, okay, tether. It connects your core to what we call the clamp loader. Okay? So part of your DNA polymerase complex is what we call the beta clamp. So the beta clamp is the one that holds on to the template strand as the polymerase does its thing. Okay, so as you can see in this diagram, you can count 3 beta clamps. Okay, 2 beta clamps attached to the core and 1 beta clamp that is attached to the clamp loader. So what's happening here? We will see in a short while, but notice that if the beta clamp is attached to the core, it's closed. But if it's attached to the clamp loader, it's still open. Why is that? Let's look at the assembly of the how DNA polymerase is oriented in your replication fork. Okay? Again, we are looking at the left replication fork. So we have here the leading strand. For the leading strand, as you synthesize the leading strand, you will only be needing 1 beta clamp. Okay? So you only have 1 beta clamp there. And then for the lagging strand, you will actually be reloading your beta clamps each time the polymerase encounters a new primer. So this ensures that while the lagging strand is discontinuous, so the leading strand is just continuously being synthesized, right? But the lagging strand is not. It's segment by segment. So this mechanism still ensures that the lagging strand can keep up with the leading strand. So what is happening here? Okay, so we have here a lagging strand, right? And we have here a segment. And oh yeah, I forgot. I think we already encountered that. We already encountered that term, Okazaki fragments. Okay, each segment of DNA, newly synthesized DNA in your lagging strand is what we call Okazaki fragments. Okay, that's each segment of newly synthesized DNA in the lagging strand. Okay, so going back, let's go back to the elongation. Okay, so we have here 1 complete Okazaki fragment. So there's the primer, RNA primer. And then here, there is an Okazaki fragment that is currently being elongated. Okay, so notice the beta clamp for this lagging strand is closed. Of course, the leading strand has no problem. We will not be dealing with the leading strand here. Okay, so the process continues, but at some point, as the Okazaki fragment here that is being synthesized nears or becomes more proximal to this complete Okazaki fragment, the primase attaches again to your helicase, okay? And it starts to synthesize a new primer here, okay? Then once it synthesizes the new primer, what happens next? So of course your primase removes itself, and then a new beta clamp gets reloaded. So it attaches sort of itself. Probably it recognizes the newly synthesized RNA primer. So the clamp loader loads a new beta clamp to the newly synthesized RNA primer. And at this point, the Okazaki fragment currently being synthesized has reached the end. So it encounters an RNA primer here. Okay, So what happens? The thing is that beta, that beta clamp will be discarded from this core because it's done. Its work is done. Okay, so it gets discarded, and now this newly loaded beta clamp will attach to this core, and then the process is repeated. Okay, so if you imagine, it's like if we go back here, so you are pulling the template strand a bit backward. Okay, you're pulling the template strand a bit backward with reference to the beta clamp here. Okay, it's pulling the lagging strand backward until it reaches the point when it encounters the next Okazaki fragment. And then it reloads a new beta clamp and the cycle continues. That is how the lagging strand keeps up with the leading strand during DNA replication. Okay, and that is um, the most important thing that you have to note of during elongation. Now, during elongation, this is where, or this is when the RNA primers are also removed. So again, this is made possible by DNA polymerase 1. So the RNA primers in the lagging strand are removed by the 5' exonuclease activity of the DNA polymerase. So what it does is, so in the lagging strand here, you have you have a nick there. So it should be nicked on both ends. Okay, so all of your Okazaki fragments are nicked on both of their termini, both ends. So if it encounters a nick, the green one is the RNA primer. Yeah, okay. So that's where it removes one RNA nucleotide and replaces it with the correct DNA nucleotide, again, reading the template strand, and so on. Okay, until it reaches the last RNA nucleotide. And that's where you have a nick this time. So the DNA polymerase 1, when it attaches the replacement of the first RNA nucleotide, replacement meaning the DNA nucleotide, it attaches to this previous Okazaki fragment, or the first Okazaki fragment, okay? Because it has the 3' OH there. It should have the 3' OH here. And then you have the deoxynucleoside triphosphate, or DNTP. Okay, so it can attach to this end. Okay, and then it reaches the last RNA nucleotide. It removes that last RNA nucleotide, replaces it with a DNTP, but now you have a nick again. So this nick is sealed by your DNA ligase. Okay, so that's what happens during elongation. So this is also happening while your replication fork is moving. Okay, so what about termination? So the question is, how does the cell know that it has reached the end? It has created, it already has the complete or almost complete new DNA strands. So if we have uh, base sequences at ORI and your DUE, or the DNA unwinding element, there is also what we call TER sequences. TER short for termination. So we also have termination sequences in your DNA molecule. Again, this is E. coli, so it's a circular DNA, and you have there several termination sequences. Okay, now if you notice here, it's called trap. Okay, the question is there a separate termination sequence for each replication fork? there is, okay, so it chances are the rate of migration of the two replication forks are not equal. One replication fork might be faster than the other, and this is dictated by the proofreading. Okay? So there might be or proofreading or some anomalies in the template strand itself, which causes one replication fork to become slower than the other. Okay? But once at least one of your replication forks encounter a termination sequence, both replication forks will stop. So either one of your replication forks encounter a termination sequence, termination stops. The other replication fork will also stop. Okay? Now, the mechanism by which this happens is still being discovered. Well, according to the textbook, but probably at this point, some information should be already known. Okay? So the thing is, the reason why you only need one replication fork to encounter a termination sequence for the whole process to stop is because if one replication fork encounters a termination sequence and the other replication fork goes on, it might have an overlap. It might do an overlap. Okay? So if one gets terminated, the other should also be terminated. Okay? Now, if your replication forks stop, what happens? Okay, so let's say both replication forks stop. Of course, you still have the termination sequence itself, which is not yet replicated. So what happens here? A different set of enzymes will complete the replication. And of course, there should still be DNA polymerase, but I guess it's a different DNA polymerase. It's no longer DNA polymerase 3 that will perform this function. Okay, so it completes the replication. And now you have what we call catenated chromosomes or catenated DNA in the case of E. coli. Each DNA, double stranded DNA here is called a catenane. So you have two catenanes at the end of a complete cycle of replication. You get two catenanes because you get two daughter DNAs, right? So the next question is, these catenanes are actually still intertwined with each other. So you need to unwind both of them. The cell needs to do that, and that is achieved by your DNA topoisomerase 4. It's a different type of topoisomerase than the one that is used at the replication forks to relieve the topological stress. Catenated DNA are also under topological stress, but it's a different topoisomerase that addresses this problem. Okay, catenane is just a term used to refer to a DNA that is intertwined with another daughter DNA. Okay, catenanes are just DNA molecules. Okay, and that's it. So once the DNA topoisomerase 4 removes the catenanes from each other, then you now have the separate daughter DNA molecules. And that's your replication. So this replication, again, takes place if your E. coli cell is dividing, actively dividing. Otherwise, you don't observe this process. But what about eukaryotes? One important thing to remember is that eukaryotes are more complex organisms, hence they should have a longer DNA sequence. In fact, most eukaryotic organisms have chromosomes, right? You don't find their whole DNA sequence in one DNA molecule. So you have these chromosomes. So because um, you have a longer DNA sequence for eukaryotes, having one origin, having one replication bubble will make replication very, very inefficient, very, very slow. So for eukaryotic DNA replication, one major difference is that you have multiple replication bubbles. You have multiple origins of replication. So imagine in one chromosome, you stretch out that entire DNA, forming one chromosome, you will see multiple replication bubbles, which is different for bacterial DNA, which is circular. So you have one replication bubble. Okay. Now, another thing that we have to consider is the timing of DNA replication with the cell cycle. So again, you only need to do DNA replication if you are creating an offspring of that cell. So the eukaryotic DNA replication machinery is more complex because it is tied up with the cell cycle. And I guess those are the two major differences. Otherwise, the process should be the same. Like you have counterpart enzymes for the helicase, the DNA B. It has a different name, of course, for eukaryotic DNA replication, but it performs the same function. And there should be more molecules involved in the replisome in eukaryotic compared to prokaryotic. Okay, because for prokaryotic DNA, it's relatively small, but for eukaryotic, you're dealing with a longer DNA molecule. So you need more Avengers, so to speak, assemble in your replisome. Okay, so that's it for DNA replication.